Good evening, everyone, and welcome to tonight's FU Alumni Webinar Series. We hope that you're all doing well during these challenging times, and we really do appreciate you joining us and sharing your evening with us tonight. I'm just going to give some reminders before we get started. I do want to take a moment to let everybody know that we are recording tonight's webinar so that we can share this with folks who couldn't be with us tonight. We have muted everybody upon entry. We should have also set the settings so that you should all be muted. And if you guys could all turn off your videos as well so that we only see our host, Dr. Avaltroni. After Dr. Avaltroni's presentation tonight, we will have a Q&A, and that will feature many of the questions that were submitted by our participants when they registered. So with that being said, and at this time, again, welcome everybody. And I'd like to take this time to introduce Dr. Karen Lewis, our Associate Vice President of University Advancement.
Thanks, Jennifer. My name is Karen Lewis, and I'd like to thank everyone for joining us this evening for our first in a series of webinars. The webinars are presented by our Alumni Association and the Office of Alumni Relations and Development. And the next webinar is going to be presented by Coach Greg Herenda, Building a Championship Team at FDU. He will illustrate how our men's basketball team was constructed into a championship program that has great regional and national exposure and impact on our university. Information about that program will be shared, including the survey right after this program, and we hope you'll join us for that. And we will have a series of other interesting topics for you to participate in. And if you have interest in other programs that we can share with you, please let us know. Tonight, our first kickoff event is our own Dr. Michael Avaltroni. He is a graduate of Fairleigh Dickinson University, receiving his degree in chemistry, summa cum laude, from our Florham campus. He went on to go to Princeton University, where he earned both his master's in chemistry and his PhD. His research was on surface modification of titanium materials to facilitate cell attachment, biomineralization, and interfacial adhesion. His research in this area has led to 5 publications in the US, 5 publications and 10 US patents of patent pending. In addition, his technology has created, in the field of study, has been licensed for the creation of 2 biotech companies. He's been teaching with Fairleigh Dickinson since 2003 and currently is the Dean of the School of Pharmacy and Health Sciences. He'll be leading our discussion this evening, which we're very pleased to have him. Reasons for Hope Amidst COVID-19. Thank you, Dean Avaltroni, for joining us and leading this hot topic for us to learn more about. Thank you again for joining us, and we look forward to the discussion.
Thank you so much, Karen. It's a pleasure. I hope everybody is doing well and staying healthy, and amidst all of the challenges is acclimating as best as possible to this new and somewhat interesting environment that we are living through right now. So I thought it would be an interesting time to be able to take a bit of a look at some of the things that maybe aren't as commonplace in some of the day-to-day mainstream media stories, and maybe is not always sort of front and center about some of the things that we should be encouraged by amidst many of the challenges of the day. Obviously, it's no secret that in a very short amount of time, life's been sort of turned upside down. And as it has been, it's been turned upside down in just about every way, whether it be economically, whether it be societally, whether it be in terms of public and personal health, in terms of school for children and workplace for adults. And one of the things that I thought would be interesting is to take a look at some things that I think should be signs of encouragement amidst some very difficult times. So first thing I do want to point out, though, is that we're fighting a serious foe. I don't want anything that I say tonight to be downplayed in any sense to make you feel like you should go out tonight and gather with a crowd of your friends. I also don't want anybody to feel as though the things that we're talking about here are in any way will usurp some of the serious warnings that have been put forth by some experts in the field, whether that be people from the CDC and the World Health Organization, whether that be by physicians and other trusted individuals, or whether that be by people who are putting out some really innovative work in this area right now. So this is something that's serious. It's something not to be taken lightly, and it's something that certainly, I think, lives up to the magnitude of much of what we're seeing in terms of the very drastic measures that are being taken. Another thing I want to point out that I think a lot of people lose sight on, and that is that I often say that this is a long game. And what I mean by that is that we're running a marathon, not a sprint. And anything that we are doing right now won't be seen in terms of the impact immediately. I think people are very frustrated by the fact that everybody in sort of the panic of the day wants to feel as though what they're doing right now tomorrow morning will make everything okay. Talking about social distancing or other actions that we're putting into place that are preventative measures are all important things for us to be doing, but it's not a cause and effect that happens in a period of 24 hours, or even for that matter, a period of, in some cases, 24 days. The things that we've done over the past several weeks will start to show impact over the next several weeks. And so sometimes what you're seeing on a day-to-day in terms of the data sometimes suggests, gee, it must not be working. But don't lose hope, because I think a lot of the things that we're doing are in fact things that will be meaningful for us going forward. And I do think that there's definitely a lot of reason for us to be encouraged by some of the things that we'll look at tonight that I believe will be some impact plays. Another thing that I will say is that when in doubt, err on the side of caution. You know, I always say that we should be doing things out of an abundance of caution. Everything we do should be, in essence, an abundance of caution, because the realities are that the risks are great. And so anything that we're doing right now, any action steps that we're taking, any societal impacts that we're having, I think are important ones to do. Whether they work to play things down to 100% of where we want to go, or for that matter, 10%, lives will be saved and people will be better for it. The last thing I will say, and this is, I think, a big one, you hear a lot of times where there's a lot of buzz around things where you're talking about something like, for example, a comparison of this to the Spanish influenza of 1918. That's a regular comparison that's made. Fair comparisons in some respects, and that certainly they are both epidemics that kind of came out of nowhere. They were both caused by viruses that were very much unseen and unknown before the time, and in many cases, because of that, there was little or no herd immunity in the sense of people having been exposed before. But I want to just point out a couple of things. When you're talking about the year 2020 versus 1918, put some things into perspective. For example, penicillin was discovered in 1928. So at 1918, although obviously penicillin is not used for viral treatments, there was no antibiotic known that was an option for any type of a interplay of some sort of a secondary infection or any sort of a concern in terms of warding off something bacterial that may have been a secondary cause for death or serious illness. Additionally, the first mechanical ventilator was discovered and made possible to be realized in 1929. So you're talking about comparisons that are really not fair comparisons. When you're looking at things from 102 years ago versus now, you're looking at technology that we have, resources that we have, the tools that we have that make this a very, very different game that we're fighting. And certainly no less serious, but we should certainly consider that many of the comparisons that are often sort of fear factor comparisons aren't valid, and in some cases are there just to kind of create a panic. So I always say to people, look closely at what you're actually seeing and look closely at the data, and the data will tell you everything. And the data in this case really tells us a very significant story. So here's an example of what I'm talking about. So when we talk about what's available for us and what's available kind of before our very eyes right now, is we look at medical technology and what we have to work with when we're treating patients, or when we're innovating to fight new diseases, or we're developing treatments or vaccines or cures. A remarkable statistic that I think is very important. They talk about the fact that in 1950, again, now 32 years after the Spanish influenza epidemic, the availability of medical knowledge was said to double at a rate of once every 50 years, which meant basically that innovation was occurring at such that there was a 100% refresh in the knowledge that was available around specific subjects and subject matters on a scale of every 50 years. By 1980, that rate was reduced to a scale of every 7 years, where you started to see this sort of full refresh of what was known and understood, the technology and the developments and all of these things that were playing before us. Every 7 years was the refresh. So you're talking about a significant exponential drop. Want to take any guess as to what the rate is now? Well, here's a little bit of a snapshot. Every 73 days, medical knowledge is said to double. Here's a little bit of a snapshot of what they often refer to as Moore's Law for medical information. Moore's Law is actually very commonly referred to around the technology realm and the rate at which technology refreshes occur. And here's a little bit of a snapshot of what you're looking at when you're talking about it in the field of medical technology. So what you're talking about, for essence of what we sort of put to comparison here, is that 5 times a year, we are learning completely new things about the subjects that we're taking on in the area of healthcare and health technology. So when you're thinking about that, think about it from a standpoint of even how we educate our students. We're educating them not to learn facts, because by the time they learn facts in a 4-year doctoral program like our pharmacy program, the refresh has occurred essentially in the essence of 20 different times. So things that were once known as the treatment, the first line, the cutting edge, in many cases now has kind of been moved to the back burner. And you see that in disease management in areas like, for example, hepatitis C. A couple of years ago, you talked about managing the disease like hepatitis C, and you talked about managing it over a lifespan or over a lifetime. Then fast forward, and all of a sudden, there's a new pharmaceutical therapy. The new pharmaceutical therapy now cures hepatitis C in a period of 8 to 12 weeks, and you put the patient into full remission. So you're talking about in a matter of a year or two, looking at the way in which you treat a disease like that affects millions of people, and all of a sudden it's flipped around and now you're looking at a completely new paradigm. Another thing worth mentioning here as we're talking about tools that we have at our disposal, things that are going on, you're looking at the fact that we can do things with computing right now that have been uncharted and unheard of in human history. IBM's Summit supercomputer, what you're looking at here is a picture of only a small subset. I actually had the privilege of touring at one point a couple of years ago, Oak Ridge National Laboratory. You're looking at what would be the equivalent of, I believe, 5 football fields filled with computers that are all networked together with the ability of this equipment to be able to perform 200 quadrillion. That's 200 trillion has 12 zeros, quadrillion then therefore would have the 15 zeros following it. If you're looking at that type of a factor of the significance in a per second calculation. What does this mean for us? It means that already the computers that we have available have been able to sequence the RNA that makes up COVID-19, have been able to determine a 3-dimensional structure of the RNA sequence, have been able to determine a mechanism of attack of the RNA sequence inside the body, have determined already the way in which the virus attacks the specific cells inside the tissue of the lungs, as well as the immunological system of the body. And it's also been used to identify 77 potential compounds that could bind to the protein that in essence will take the spikes that you're used to seeing in these pictures that you see everywhere, that will essentially render them useless and make them unable to attach to cells, and would ultimately lead to potential treatments and or vaccine remedies. So you're looking at already in the short time that we've had knowledge about this disease, we're looking at the fact that we've already been able to sequence it, understand it, know how it's metabolized and what happens inside the body, how it reproduces, how it acts, how it triggers immune response, and then ultimately 77 potential different ways that we could solve the problem by synthesizing compounds that could potentially treat the virus by shutting it off and not allowing it to replicate in the body. So when you look at that, that's happened in a matter of about 15 weeks, plus or minus, probably much less than that in essence, because in fact, what you look at is, in fact, most of the research didn't start picking up around this subject area, particularly in the US and Europe, until probably late January. And in fact, the CDC and the FDA did not really authorize any types of research going on in this area with any substance until that time, when they kind of gave the charge to people to start looking further into this. So we're looking at, in a matter of less than 2 months, we already have all of these answers before us. It's a pretty remarkable turnaround, and it's a real testament to the fact that we have so much intelligence, both human and then artificial, simply at our disposal. What does it look like in terms of how does it manifest itself? You've got companies right now that are working on this disease and how to potentially solve problems in a way that has been sort of unprecedented as never before. Here's a little bit of a snapshot here. There are currently 39 potential vaccines that are either in preclinical or clinical phase right now being developed. In fact, you may have seen recently they administered the first vaccine treatment to a human. That happened about a week or a week and a half ago. It occurred administered by a pharmacist to a patient to test out what in fact happened in terms of immunological response. And remember, what we're always looking for here is two things. The first is it safe? The second is it effective? So these clinical phase 1 trials will determine that in a small sample before that sample is then scaled up. And then ultimately, if it looks good still, then moved to a manufacturer. What's important about these vaccines, in fact, which you'll see here as evidenced and sort of listed in this potential table here of leads, is that in fact, most of the vaccines that are going for the first trials are designed in a different way than vaccines have ever been designed before. Most vaccines are designed to be either using live attenuated virus or in some cases a killed virus. In this case, they are actually using specific sequences of RNA that are the specific binding sequences that they've identified that would be the critical pieces to develop immunological response. The reason that's significant is that has long-term ramifications, that if we in fact develop a successful treatment of a COVID-19 vaccine, you can then back architect this style of a vaccine delivery for treatment for other viral diseases. For example, rather than the somewhat unsuccessful, some years, flu vaccine, you may be looking at now flu vaccines that can be developed much more effectively to have both a longer term staying power, meaning they don't need to be annualized, but also much higher effectiveness in terms of seeing the outcome that you want. Additionally, other viral diseases like, for example, Zika virus, dengue fever, West Nile virus, HIV, other types of things now can adopt this technology if we see it work here. And suddenly now you potentially have platforms for vaccine delivery to cure a lot of significant diseases that are affecting a whole lot of people. Here's another bit of a snapshot. One of the things that I find fascinating is that I'm putting these slides together literally in real time. Up until this evening, I was editing things because things are happening so quickly that you're seeing things actually before our very eyes move on and off these tables based upon clinical trials, based upon new studies, based upon things that are happening in real time before our very eyes. So this is a small snapshot of some of the potential leads that are in either phase 4, which is basically ready to deliver, phase 3, phase 2, or phase 1 trials through FDA and in some cases also international approval cycles. You'll see what's interesting here in this table is the cross-section of companies that are working on these particular drugs. You'll see lots of different companies represented here. Some of them your big players, some of them startups or smaller players, some of them affiliated, for example, with universities and other institutions usually using intellectual property there. And another interesting thing is you'll see very different treatment modalities here. You'll see some of them are specifically designed to be inhibitors. Some are designed in some cases to be antivirals, anti-inflammatories. Some of them are treating the symptoms. Some of them are treating the virus or attempting to get the virus before it begins to replicate. And there's different structural approaches to taking on this problem. The big factor here is that what we're seeing right now is that there are a lot of different things, but there are a lot of different things, even more importantly, attacking in a lot of different ways. And that, of course, lends itself to a potential buildup of having multiple modalities of treatment, not just simply one, which is certainly to our advantage as well. Now, here's the thing that I think is going to be one of the very, very near-term game changers. You've heard all along from a lot of people who are speaking about this disease that the answer here is test, 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 right? Obviously, that's the shortest term method that we have to being able to control this epidemic. And the reason for that is that the big concern about why there's been so much significant spread of this disease, unlike so many others, is what they're concerned about here is the very, very widespread asymptomatic spreading of this disease, both by people who are asymptomatic before the disease starts in terms of the symptoms, and also for people who remain asymptomatic across the entirety of the disease's manifestation in their body. So what does that look like, or what does that mean? There are, as you've probably heard, a significant number of people who would in fact test positive if tested, but are walking around with minimal or no symptoms at all, and as a result are potentially spreading this virus without any knowledge. If we can develop more efficient and more effective testing, you have the ability to then create a structure in place where people can determine their test status and know whether or not they need to self-quarantine, not on the basis of symptoms, not on the basis of a diagnosis through some sort of a cough or a fever or something else, but simply on the basis of an abundantly available positive test that happens in real time. And so what you're looking at here is you're looking at a significant number of these tests. Most of them are what you would look at as PCR, or polymerization chain reaction tests. Those are diagnostics for determining presence of the virus inside a person in real time, depending upon the result longitude in terms of how long it takes. However, another important factor here, which is on the cusp of being developed and approved right now by the FDA, is in fact also serological testing. What does that mean? That means testing for the purposes of getting a knowledge base of whether or not somebody has been exposed by testing their blood for, in fact, the presence of the viral factors through a titering approach. Much like you could determine whether or not, in fact, somebody is immune to measles or mumps or rubella through titers in the blood, this can now be done in the case of COVID-19. That has multiple critical important pieces to it. The first is if you can determine people are demonstrating immunity, those people can go back into society in a sense of being healthcare practitioners who no longer have to concern themselves with exposure risk. You can put these people back into the society to be able to once again earn a living, knowing that they're not a high risk factor of either spreading the virus or getting the virus. So once you have the ability to do these types of modalities of testing, you can determine, in fact, how widespread the presence of COVID-19 is, and is there, in fact, a much wider audience of people who are immune, who have had the disease but have never shown or displayed symptoms, or in some cases have displayed mild symptoms several months ago and thought it was just simply the common cold, or thought it was a bad case of an upper respiratory, or thought it might be a mild case of the flu. So that's going to be a significant factor that's going to be a huge game changer as that is developed. But here I want to show you is what I think, and many others think as well, is going to be a significant game changer for us immediately. And I'll explain a little bit about why I think it's so critically important. There are multiple components here. So Abbott this week, actually a couple of days ago, got an FDA approval for a rapid COVID-19 test. It determines a positive in a matter of 5 minutes and a negative in a matter of 12 minutes. And it's effective at a lower bound of 92% and an upper bound of 99%. So almost no false positives or false negatives here. Now, the reason this is such a big deal is a couple of factors. First of all, Abbott is committed to the fact that they believe they can put at least 100,000 tests a day into the hands of medical practitioners in the country. The other thing is that there are 13,000 of these machines that are shown here in the picture that are currently deployed at healthcare units across the United States. So what that means is that these folks already have the modality to test. They simply need the testing strips and solutions, and they can begin hitting the ground running and doing tests thousands at a time immediately. So if you're looking at 100,000 tests a day entering the supply chain and entering it immediately over the course of 4 weeks, that means we are looking at, in essence, 2.8 million tests being available to be done point of care with the machines already deployed in hospitals and urgent care centers and other physicians' offices where they're currently being used to do similar tests for influenza A and influenza B and for rapid strep. So you're talking about not some sort of a new modality that's going to take a long time to roll out. This is something we can deploy on site and on the fly right now. And again, the more people we know who are testing positive, the more we can be deliberate about putting effective quarantine into place and then building back in our society parameters around what constitutes quarantine and what constitutes the build back to beginning to create some normalcy back into society, particularly around getting people back to work, getting people back to school, and getting people back to life, which is a critical component to obviously this disease and why it's so societally damaging. Another thing that happened this week, and another potential lead that I think is a very exciting one that also got very, very widespread ramifications well beyond just COVID-19. This week, Mount Sinai began requesting for those who were recovered from COVID-19 to begin donating blood and plasma for the purposes of treatment of critically ill patients in a clinical trial that's being done in real time, where they are treating these people with what they call convalescent plasma. That is the blood and plasma of people who have recovered from COVID-19 and are now healthy, where they are basically using this to try and trigger an immunological response that would allow people essentially to begin to have kind of a boost to their immunity to begin getting an assist from this treatment towards helping them to lower their own immunological response, which often causes a lot of the side effects of inflammation and other things that are the major problems in why people are needing to be hospitalized and in many cases going into respiratory distress. But you're also seeing that this type of a treatment, which actually has seen some success, you'll see the publication that's cited here from March 27th from 3 people in China who actually were doing this with real-time cases in the sort of epicenter of the outbreak initially and saw significant results of improvement and in some cases a very, very significant reduction of viral load over a course of, in some cases, as short as 48 hours. So you're seeing breakthroughs here that could potentially be a huge game changer in terms of getting to these patients early, particularly by looking at those who have a high risk profile for respiratory distress, usually through people who are presenting with some other comorbidity, something like diabetes or heart failure or some other disease state that puts them at increased risk. But additionally, what you're going to find is that this, again, has long-term treatment ramifications for a lot of other disease states as well. So the question becomes here on a lot of these things, can what we're learning about COVID-19 help to solve not just this problem, but help to solve a lot of other medical problems that have been plaguing us for many years and have caused a lot of tax on our healthcare system and a lot of people who have ended up with poor outcomes and in some cases death as a result of these disease states. Now I'm going to talk a little bit about some of the things that you've probably heard, maybe in some cases kind of ad nauseum. You're hearing a lot about, and a lot of sort of the back and forth in the political footballing about this sort of two-drug treatment, what they refer to as a kind of cocktail, hydroxychloroquine or chloroquine as well, mixed with a first-line antibiotic, zithromax or azithromycin, that has seen a dramatic increase in recovery time and a dramatic reduction of viral load in a very small segment of patients where a clinical trial was done in kind of a very, very, I would call, dirty way, you'll just say. A lot of the concern here has stemmed from the fact that the things that we're seeing right now don't follow often the best practices of what you would think of when you're running clinical trials. And the reason is that we're trying to do a lot of these things on the fly in real time amidst a critically ill patient population who have some desperate needs that we need to address immediately before they end up in serious respiratory distress and in many cases at death. So what you're looking at on the left is in fact you're seeing what was the initial study that triggered a lot of the interest around this chloroquine azithromycin treatment plan. And what you're seeing there was, in the green line, a significant reduction in the case of percentage of positive samples collected, showing that there was presence of virus when these two drugs were administered, relative to the control, which is seen in black, and the hydroxychloroquine only line, which is seen in blue. Now, it is long known, in fact, that these two drugs actually are shown to significantly reduce inflammation. Hydroxychloroquine and azithromycin both have those properties about them, as well as other properties that they're often used for. Hydroxychloroquine has been widely used as an anti-malaria drug, developed in the 1930s and widely used for that, but also more recently has been used to treat lupus and rheumatoid arthritis, largely because of the anti-inflammatory factors. Azithromycin has been a first-line antibiotic used for many years, and it's a very, very inexpensive remedy here that is also very abundant. The nice part about this potential drug regimen comes from the fact that it is both inexpensive and very readily available, because both of these have a significant generic supply chain available, and both of them are very, very much cost-effective treatment plans which could be widely dispersed to large populations. Recent publication that came out here, in fact, over about I would say 3 or 4 days ago now, you're looking at, on the lower right-hand corner, a paper published out of France that in fact showed a significant progress of the same in the process of doing this on a more regimented clinical trial, where you were seeing a significant impact here as we looked at the overall result of trying to treat these patients with these two drugs. Now, the thing I will tell you is that there were a lot of people who were looking at these papers with a lot of questions, because a lot of the normal protocols that were followed in these types of clinical trials were kind of bypassed here. So there is some concern about how good is the data and how valid was the study. But ultimately, the thing I will point out and tell you as well is that in many respects, we have to understand here that we're in some uncharted waters. And the reason is the normal protocols of developing a clinical trial and taking months to do studies and being able to develop a course of study and evaluation in a really sort of scientifically relevant modality requires us to wait out ultimately what we're seeing right now, which is this epidemiological crisis before our eyes. So a lot of the corner cutting that's happening is the result of people trying to get to market very quickly with things that could potentially help and ultimately give us the outcome that we're looking for. So it is definitely, I think, something that we want to look at with definitely a keen eye, but certainly there is hope here amidst these studies, and definitely something for us to look further at. What I will tell you is that there are some concerns, particularly because a lot of the people who are high risk in these sort of parameters of using these drugs also often present with comorbidities of heart failure or other cardiological issues. There are some significant cardiac side effects to both of these drugs that need to be evaluated very carefully. Another thing that's important is that we're starting to see already that there are being some very tight restrictions put into place, because you're already seeing drug hoarding, where people are writing prescriptions, physicians, for family members and friends for 100 hydroxychloroquine to be dispensed. So people have it on hand, either to take it prophylactically, which there's no evidence to suggest at this point is a good idea. And there's also a lot of people who are hoarding it in the event that they need it, knowing that there's going to be a shortage in the supply chain. So a lot of things here are concerns, because this became such kind of a media buzz that all of a sudden everybody wants to know more and wants to understand more. One thing that's also important that I will point out here amongst all of the things that we're talking about, and that is that clinical trials are also typically not run on critically ill patients who have comorbidities of diabetes and heart failure and others, which are the exact population we're trying to help here. So a lot of the things that we're doing actually run counter to best practices of clinical trial design. The very people who you would normally exempt from a study because they would not be good subjects in a clinical trial are in fact the very ones that we're trying to help with these drugs. And so we're almost in many respects kind of working cross purposes here to try and do one thing apart from what we know is the best thing scientifically. And that's a bit of a struggle that everybody's trying to navigate and get through. So as we kind of look further at things here, there are other treatments as well that are showing significant promise beyond this one. In fact, the one that probably has shown the most encouraging signs is one that's a product that's put out right now by Gilead, the makers of also a very significant number of other drugs that were antiviral. One of them, actually the most well-known, has been a treatment for hepatitis C. This remdesivir is in fact an antiviral agent which has shown significant impact to have a very, very, I think, positive effect on a number of other single-stranded RNA-based viruses, which is in fact what COVID-19 is as well. They have shown significant impact on treating SARS, the virus from 2002, 2003, the Southeast Asian virus, and then MERS, which was the Middle Eastern version, which came out in about 2006 and was shown as a potential treatment for that at that time as well. So what they've seen here is they've seen that there's been significant impact, largely because the viral mechanism of action is to typically get into cells and begin replicating by attaching into cells with the RNA backbone of the virus. What is known is that this particular treatment can essentially replicate or resemble the viral RNA and attach to the RNA itself and create a blocking mechanism, which in fact blocks the RNA from replicating inside the cells and blocks those cells from reproducing, which is the large way in which viral onset occurs so rapidly and so dangerously. So we're seeing a lot of promise here because the mechanism of action of things like SARS and MERS and other respiratory diseases that have evolved before, we're actually seeing a very similar mechanism of action here, and this drug has been very effective there. So a lot of positivity potentially on the horizon with things that are going on here. Studies are underway right now in a number of different areas, going and taking on clinical trials in this particular drug class. One thing that's also happened that is a very important development is that the World Health Organization has begun now to be sort of a clearinghouse of aggregating different nations' clinical studies and clinical study teams so that you get more of an economy of scale of these groups working together. And you see a lot of things happening now and transforming the research in a much more rapid scale, which is a huge thing as well. You're seeing another drug, Kevzara, which is Sirillium. That is in fact a different mechanism of action drug. That's a monoclonal antibody put out by Regeneron. And that has actually been seen to have an effect at stopping the body from triggering an immune response that leads to significant inflammation that causes this respiratory distress and an essentially an attack on the body's own cells. So what you're seeing is that this monoclonal antibody, which is actually a method of treatment used very commonly in cancer and rheumatoid arthritis and other disease states where monoclonals are used to essentially block a lot of the regeneration of cells and the immune response there. This would be a similar type of drug delivery where you're taking this mechanism of action and you're blocking this cascade mechanism from occurring in the body so rapidly and potentially reducing inflammation and reducing what really happens in the high risk factor of the viral kind of onset, which is the body essentially through the virus's integration into the cells beginning to attack itself, and in some cases so rapidly that it puts the body essentially into a form of kind of a shock. And then there's another drug that's been out for a while, Avigan, which is Favipiravir, which is the name of the drug generically. Also an antiviral, which you'll be able to determine the VIR on the end of the drug name implies that. And that is in fact also a drug that has shown significant activity against influenza. This is a drug that has been widely used in Japan, put out by Fujifilm. And in fact, it's been used with treatment in influenza A and B and shown to have effect there. West Nile virus, dengue fever, and actually a couple of other antiviral mechanisms for the purposes of treating viral illnesses there. And the thought process, again, a similar mechanism of action and a similar approach to being able to deliver this drug in the hopes that it would shut down the virus from being able to replicate so readily in the body. So these are all things that are being studied right now, and in real-time data is being collected. And in many cases, they're authorizing compassionate use for these drugs to actually be delivered to patients in dire need who are in critically ill situations in the United States right now. So you're seeing that in some cases, we're not waiting for the data. We're actually administering these through compassionate use with the purposes of hoping that through this compassionate use, we can also learn a lot more about them while we're potentially saving lives. Just another thing to point out that's got another reason for potential encouragement here. Recent publication also hit about a week ago, or maybe at this point in 8 or 9 days. Scientists at MIT did an extensive study of the virus under different conditions of environment and actually found that the most common infection temperatures occur at a rate between 37.4 and 62.6 degrees. That should be Fahrenheit, my apologies. 37 and 62 degrees Celsius would be much warmer than any of us would be sitting through right now. 37 and 62 Fahrenheit, and that would actually be very comparable to what's seen with a lot of viral profiles. And that is that viral profiles are seen not to necessarily be reduced overall in abundance, but there's a much greater tendency to reduce the actual infection rate in warmer and more humid climates. So you're actually looking at, in a lot of cases, when you look at the map of infection rates, you're seeing that most of the predominant outbreaks have occurred in these sort of colder climates right now. And it's well known about influenza as well, that influenza is always thought of as more of a cold weather disease. Not even known very well as to why that is, other than the fact that the theory behind it is that on warmer, more humid environments, the virus has a more difficult time adhering to the cells in the body that actually makes this sort of possible, this infection possible. Again, there's a lot more to be studied and known about this, but what is known is that this occurs readily in RNA replicating single-strand viruses, and COVID-19 being one of them. The assumption is, and the preliminary studies, that in fact you do see that as weather gets warmer and more humid, infection rates go way down. So the hope for us is that right now, time is also on our side, and that the season that we're approaching, and where much of the infected world right now is approaching, will lend itself to being able to hopefully have a reduction overall in infection rates. All of these things ultimately will help us to slow time as we begin to develop better tests, better treatments, and ultimately a vaccine. So, a couple of things also that are more on the front of what we are doing right now as a society. You hear very regularly discussion all the time about flattening the curve. And what does that all mean? We can talk about it for hours and explain it, but I'm sure you've seen it and heard it on the news. And ultimately what we're doing right now is we're trying to reduce the overall infection rate by taking an approach of social distancing or quarantine. And the way in which that works is that ultimately when you're looking at an infection, you're looking at a rate of transmission, what they oftentimes refer to as an R0, that differs based upon what different type of infection it is. Some things have a very high R0. Things like measles, for example, have an R0 somewhere in the order of about 11 or 12. Other diseases have a much lower R0. Influenza seasonally has an R0 of about 1.4 to 1.8, depending upon the season. R0 is simply summed up as the number of people that one person can infect at a given time. So for example, one person coming in contact can infect 12 people with measles. One person coming in contact will infect 1.5 or so people with influenza. And it's thought that COVID-19 has an R0 somewhere on the order of between 2.2 to 2.8. Different studies in different times have suggested a wider spread of numbers even than those, but that's the thought of based upon the preliminary epidemiological data. With that in mind, the goal at this point is to try and get that R0 down below 1. And the reason is very simple. What that means then is that the rate of infection is such that one person is no longer infecting the next person. They're infecting just a fraction of those people. The lower the number goes, the more effective the spread will, the distancing will be so that the spread is not as wide, and that the slowing of the infection rate allows us to then control the medical system and the tax on it, to control the rate at which people need to pursue healthcare, and also control ultimately, and in some cases shut or entirely shut off the spread of the disease. Interesting study, or an interesting discussion is that in fact, if the entire world were to have self-quarantined for 14 days, this and every other virus would completely go away. Obviously, that's not practical, so the goal is to do what you can. But if everybody would have taken an approach from day one to say, this looks bad, let's sit home for 2 weeks, all of this would have been gone in 14 days. And again, obviously, that's sort of an optimized reality. But in fact, the better we can do with this distancing, the more effective we will be at being able to manage this spread and ultimately also manage our healthcare resources. So here's a little bit of a snapshot of some of the syndromic surveillance data made available by New York City. Table on the left, in fact, is what they would call influenza-like illness. The table on the right is respiratory. These are, in fact, emergency department intakes of these people going and seeing a healthcare provider in the emergency department on the left, presenting an influenza-like illness, and on the right, presenting some sort of a respiratory distress. That can be anything from just a cough, to having difficulty breathing, to having shortness of breath, to being cyanotic from not getting enough oxygen and having a low pulse ox, and so on and so forth. But what you can look at from both of these curves is the interesting part on the right. And that is what you will see is a significant turn down over the past 5 days of the intake of these people hitting the healthcare system in the 5 boroughs of New York for either influenza-like illness or respiratory distress. That's significant because what that ultimately means is that that is about 14 days past the window of when social distancing was promoted and put into effect by the governor and mayor of New York. So what that ultimately means is we are seeing already a significant impact. Now, I do want to point out that one thing that's important that you have to keep in mind is that, in fact, you don't want to go down the road of looking too much at the data about death rate and about positive tests. Death rate is a much slower process. So we're going to see that impact hopefully turning around in probably another 7 to 14 days. And the reason is that just because you were admitted to the emergency department doesn't mean you're near death. The emergency department may mean an admission and a move to the ICU and then days on a ventilator, then ultimately leading to death. The other part is the testing rate. When you're seeing more and more new tests that are positive being reported, that's normal and understood because the effect right now is that we're increasing the number of testing exponentially. So the way I always say it is, think about it this way. If I test 10 people and one of them tests positive, I've got a 10% rate, but I've only got one positive case. However, I've only tested 10. If I test 10,000 and I get 10 tests positive, I've seen a 1,000% increase, but my rate of infection goes from 10% to 0.1%. So numbers can be very deceiving. So don't fall into the trap of saying, oh, there's more cases, things aren't working. Things are working when we're measuring the tax on our healthcare system and seeing it decrease. And you can look at this actually as well in Washington State. Here's an example using those R values, which you can see are actually down approaching 1 right now, which is exactly where we want them to be or below. But you're seeing that as a result of putting into effect a slowdown of the system has occurred as we put social distancing and quarantine orders in effect in Washington State. And here's a snapshot of looking at a similar set of data of COVID cases in Italy. And you can see significant drops there. And in fact, today it was reported that even a more significant percentage drop of positive cases in hospital admissions occurred in Italy as well. So we're seeing things happen and we're seeing things work. And that's an important factor for us. Last thing I want to point out just quickly that I think is interesting, that's one more thing before I wrap up and take some questions. There's some other interesting data that's at our fingertips right now that's really on the precipice of breaking out, and I think in many cases really transforming the way in which we practice healthcare as well. Here's a little bit of some smart technology use as a future of predictive modeling. So this was actually produced by Kinza, which is a smart thermometer manufacturer. And what you're seeing actually here, this I took as a snapshot of data that I collected from the website today by collecting what looks like the cumulative atypical illness rate based upon an increase in temperature readings that are fed to the smart thermometer into a smart device and then back up into the cloud. And what you can see that's fascinating here is that in fact the epicenters that have been talked about with the increased number of cases in fact show in real time exactly what you see as elevated temperature readings on these smart devices. And in fact, you can also look at trend lines. Here's a snapshot, which is an encouraging one, at a 7-day trend in illness based upon these increased temperature readings, which you'll see shows decreases across the United States based upon where they were 7 days ago. And you'll see less significant decreases in areas where you're starting to hear pockets of increased spread that are starting to take place, places like Texas, Louisiana, and elsewhere where they've talked about potential outbreaks that are the result of things that they haven't done or haven't done properly yet. And here you can even get hyperlocal. This is a snapshot looking at Morris County, New Jersey, and looking at the zip code home of Florham Park, and looking at, in fact, what the data looks like there in the tri-state area as we look at, in fact, these pockets. But another encouraging sign, again, if you look at the trend line on the bottom, you'll see a significant elevation that was there several days ago from what would be the observed normal. And we're starting to see that data line now trend more to what would be what they call the expected trend line. And the hope is that it continues to drop, showing that in fact there's a decreased number of people presenting with an elevated temperature, one of the classic symptoms of COVID-19. And as a result, we hope that that means in fact that there are less people presenting with new symptoms. So all this to sort of tie it up, what are the takeaways here? I say ultimately there are a couple of them that I think are significant. The first thing is that we're seeing global innovation changing the way in which we learn, develop, and advance technology. And this is in fact to address what we can, without question and without argument, say is the most serious public health problem that we've seen in the past 100 years. And we're seeing this happen, and we're learning from this that in fact we should never be the same again. One of the things that I hope would be the outcome of all of what we're seeing here and what's going on here is that we need to move away from practicing sick care and addressing things in crisis management time, whether that be with how we secure personal protective equipment in our hospitals, whether that be how we develop new technologies to treat diseases, whether that mean how we develop vaccines and other treatments ahead of when these problems start. There were advances in vaccines around RNA viruses back in 2003 and 2006 when SARS and MERS were the topic of the day. What happened? All of a sudden, those things go away, and with them goes the desire to do any research in that area because there's no money to be made there. So then we see drugs like Kybella, which is a drug that treats double chin, which now all of a sudden comes front and center, and they're starting to do advertisements around. While it's very nice to have cosmetic treatments in the research pipeline, the question right now that we should all be asking is, where would we have better spent our money? Treating and addressing the concerns around new viruses that could potentially be societally changing, or remedies for treatments that affect a few people or affect how people look? And I think these are important things that we have to ask within the healthcare industry and within pharma and biotech industries. How do we better resource ourselves so that this never happens again? We need to invest in public health. We need to use what we learn to take on new challenges. And we need to realize the value of healthcare and health professionals, which we are seeing before our very eyes, put themselves at risk so that they can solve this problem and that they can treat people in real time with very limited resources and very intense conditions. And then the last thing is we need to figure out how we continue to redeploy our resources to improve our health outcomes. How do we better use the people that are out there, physicians, PAs, nurse practitioners, pharmacists, and others, to really be making an impact in ways in a given day. What I will tell you right now, we'll have time for some questions. I'm happy to answer those questions. If you have more questions, want to know more, want to talk more, please feel free to reach out and talk with me. You can contact me by email or by telephone at any point in time, and also a link to some of our programs in pharmacy, in public health, in the physician assistant program, which will be coming on board in 2021, as well as our social work program coming on board in 2021, and our occupational therapy doctorate also coming on board in 2022. And so I look forward now to answering some questions, and I thank you for your time and your attention, and hope you've enjoyed learning a little bit about where we're going, and hope you're encouraged by a little bit of what you've seen here.
Thank you so much, Dean Avaltroni, for such an informative presentation on COVID-19. No doubt your knowledge and research has brought understanding and hopefully hope for all of us among COVID-19. So thank you very much. We're going to go ahead and start with the questions that were submitted. The first question is from Kimberly Yee. She's an alumni from FDU, class of 2008. And her question is, has there been a set cure of the virus? I have heard that Indian and African populations have not been hit by this, possibly because of the anti-malarial medication.
So you're definitely seeing reduced rates in certain countries. India now is starting all of a sudden to see an uptick that's been significant. In fact, I believe that is a 3-week quarantine that's been put in place for India at large, where now 1.5 billion people are under essentially a kind of a lockdown order. Africa is starting to see some uptick as well, but definitely less of an outbreak that you would see there than you probably would expect, particularly based upon populations. There are a couple of theories. One of them is that people are speculating, in fact, that the anti-malarial agents that have been administered there may have some prophylactic effect. Nothing has been concrete to determine that. There's also a question as well as to whether or not the climate or weather also has a factor, as I alluded to before, knowing that warmer, more humid conditions tend to be less conducive to viral replication and viral infection. So that's unknown at this time. The one thing I will tell you is that as this sort of continues to be more well understood, we're going to get a lot of answers here, and we're going to better understand what in fact are some of these sort of unknown at this point parameters that may be factors that are hidden within a lot of the data that we're getting.
Thank you. Another question we have is from William Heaney. He's also an alumni, class of 2012. Why does this virus seem to hit a younger demographic harder here in the US than it has hit in Europe and Asia?
So there's a couple of things that I think are at work here. One of them I will say is that if you look statistically, in fact, I believe there was actually a paper just published this morning, I think it was from the New England Journal of Medicine, that actually talked about the fact that they are expecting, although the mortality rate is significantly higher than influenza, they're believing it to be somewhere on the order of 0.6% right now, which is much lower than the initial projections. And they are also showing that that mortality rate is significantly curved and bent very heavily in those 80 plus. So there are, in fact, younger people being affected. But in fact, you're seeing a lot of one-off stories that are picked up in the media that sort of exacerbate what the actual data suggests, meaning that you're seeing a lot of those brought to the forefront because largely they're so anomalous. You know, an 85-year-old person passing away is obviously less of a story than a healthy 32-year-old person. So a lot of that kind of seems much more exacerbated than actually the data suggests. But there's also some other theories that people have as to why potentially it may be hitting harder in certain populations here as well. There are some interesting studies preliminarily, again, not much data yet to be able to mine, around the questions and factors of smoking and vaping, as well as potentially marijuana use as potential causes for some of the respiratory tax that may be placed on younger respiratory systems that could potentially then lead to compromised respiratory function. Again, nothing concrete yet in the data, but just some interesting things to watch. Are there things that we've learned about vaping that we were concerned about a year ago at this time in terms of some of the younger people seeing some real respiratory issues as a result of that now are at play when we're actually seeing them infected by a respiratory illness? Still more to come, but a theory that's potentially worth watching.
Thank you. Our next question is from alumnus Stephen Grumbach, class of 1977. And his question is, while supermarket shopping, droplets versus aerosol, and what is the infection risk in the aisles?
So yeah, that's everybody's question. I actually have full disclosure. I was at the supermarket today. I did wear a mask, I will tell you, largely because I'm watching actually, in fact, new research from the CDC that is suggesting that they may come out and now at this point say that mask use may be actually advisable. Thought process on mask use is very interesting in that most of the time masks are used to protect the person who's on the other side of the mask rather than the mask wearer themselves. So in fact, most of the time what you'll notice is that a mask is given when somebody's admitted to a hospital or an urgent care center if they have flu. The person who's infected is made to put on the mask. Everyone else is not wearing the mask. The thought process is that it's droplet containment of the infected person. But the new mindset that the CDC may be moving towards is in fact thinking about that if I wear a mask and you wear a mask, if only one of us is infected, then we're both protecting each other. So if we want to get back to more normalcy sooner, then mask use may be a prudent thing, largely because you'd say, well, ultimately, if we're all protecting each other, then it is kind of the public's health. The reason they haven't moved more rapidly at that rate is because they know immediately that when they say that, they'll be hoarding. And there'll be then a shortfall of PPE available for healthcare providers because everybody will be going and trying to buy their own black market N95 mask. And so that's the concern, is that when any recommendations made, people panic and they jump. The realities of what's known thus far and what's been suggested, studies thus far have suggested that it is in fact the aerosol that is the major concern. That the reality is that the virus is traveling in saliva and droplet containing salivary contact. So you're seeing that sneezing, coughing, and that type of thing, and in fact, even talking and spraying in that sense is the risk factor. There is nothing at this point that suggests, although again, it's always prudent to be extra cautious because you don't know what research will point toward. There's nothing that suggests that you're seeing long-term remainder of virus that's active, live, and infective. They often talk, and I always caution people, they talk about finding, for example, virus on or viral components or RNA on cardboard after 4 days, or in a cruise ship after 14 days. There's a big difference between finding traces of RNA and actual virus that would be able to infect you. That is not the same thing. So be cautious when people talk about that it doesn't trigger fear, because oftentimes when you see traces, that doesn't necessarily mean if I touch this and then touch my face, I'll be immediately affected. So a lot of this is still to be determined, but definitely at this point right now, the biggest concern is close proximity to people who are ill or even asymptomatic and their salivary contact.
Thank you. And Dean Avaltroni, we have time for just about one more question here. So I'm gonna go with this question from alumna Novlet Hammond, class of 2004. What are your thoughts on how this virus will impact healthcare moving forward, and what practices need to change? Excellent last question.
Great question, and I think a great way to sort of sum it up. So I alluded to a little bit in kind of my conclusion. The reality is that if we don't change, this will happen again. And the reason I can say that with good confidence is that it may not be something that happens to this magnitude. But the reality is we can look at trend lines over the last years, and we've seen these sort of blips happening. And we know that what we've done ultimately is we've practiced healthcare in a way that kind of, in many respects, doesn't prepare us well. And what I mean by that is that a case in point is that we knew that there were these outbreaks occurring in China, and we didn't, as a society, do anything to put parameters in place to protect our healthcare workers. We didn't order surpluses of N95 masks and gowns and gloves and other things, largely because the way we practice healthcare and the way we ultimately kind of deliver healthcare is that we want to only spend money after we have to, and we want to only invest after there's necessity. So the reality is a hospital executive would have been sort of laughed out of an office building if they would have said, I want to buy 1,000 extra ventilators and I want to buy a stash of 50,000 extra masks just in case. There's no budget money for that. There's no reason for that. I think what we're learning right now is that there needs to be more invested in prevention and more invested in awareness and more invested in prevention of the next big thing. And I think the other thing that I would say to take home in all of this, we need to also empower our healthcare professionals. What you're seeing right now is that every professional inside healthcare right now is making an impact because they've largely been unfettered and unrestricted from practicing at the top of their license. And what I mean by that is that people are no longer saying, oh, you're a pharmacist, you can only do this. Oh, you're a PA, you can only do that. Oh, you're a nurse practitioner, you need to ask this. This has become a lot like practicing almost on the battlefield right now. And so a lot of what's happening here is there's autonomy and there's an expansion of the scope of practice that is a good thing that will lend itself to better health outcomes well after this disease has come and gone, and well after the next one has come and gone too. So the hope for this is that we never go back to the same way again. And we see our healthcare practitioners authorized and empowered to be able to practice at the type of their license and to be able to do the things that they should be doing and that they can be doing if we would just get all of the barriers and restrictions out of their way. The other thing I would say as a take home is make sure we're showing appreciation to those people who are truly risking their lives to do what's right for us. You can't understate, and as I've spoken with people out there on the front lines, what things are like right now, and the realities that these people who have spouses and children and lives are sacrificing. And that's not just also the physicians and the nurses and the pharmacists, it's also people who are on custodial staff and dietary and elsewhere. These people are going into the war zone every day, and they are doing what's right to protect the public and to make sure this is a best outcome for the people they're treating. And that's a wonderful thing.
Okay, so just to say thank you once again, Dean Avaltroni, for providing us with your presentation for our first alumni webinar series tonight. We really appreciate your knowledge and your input on this topic. And I want to say thank you to everybody for attending. Please keep an eye out for an email. We will be sending you an email with an evaluation on tonight's webinar so we can get some feedback. and make sure that we're providing good programs for everybody. So please check out for that. And thanks again for joining us tonight. Stay safe and stay healthy. Everybody have a good night.